Yeah. So here, this is a thin membrane that vibrates as the sound waves hit it. Think of it as the head of a drum. This membrane is attached to the first in a series of three small bones collectively known as ossicles. The eardrum connects with the hammer, which is connected to the anvil, which connects to the steer-up. The vibration of eardrum is transmitted by these three bones to the oval window, a membrane very similar to the eardrum. The oval window membrane is attached to the cochlea, a structure shaped like a snail's shell filled with fluid. As oval window vibrates, the fluid moves. The floor of cochlea is the basilar membrane. It's lined with hair cells connected to the organ of Corti, which are neurons activated by movement of their hair cells. When the fluid moves, the hair cells move, and transduction occurs. The organ of Corti fires, and these impulses are transmitted to the brain via the auditory nerves. And next one is pitch series. So the description of the hearing process above explain how we hear in general, but how do we hear different pitches or tones? As with color vision, two different theories describe the two processes involved in hearing pitch: place theory and frequency theory. The number one is place theory. Place theory holds that the hair cells in the cochlea respond to different frequencies of sound based on where they are located in the cochlea. Some bend in response to high pitches and some to low. We sense pitch because the hair cells move in different places in the cochlea. And this is our ear. So this is external auditory canal. And then this is external ear and middle ear and internal ear. And there is an eardrum. This one, eardrum. And here, there is a, like here in the sounds, inside the eardrum, there is an oval window, this one. And eardrum and oval window, there is a hammer and anvil, and then steer up shapes here like this. And there is a semi-circular canal. And this is cochlea and auditory nerves. This is our cross-section of the ear. So, and the other theory is frequency theory. Research demonstrates that place theory accurately describes how hair cells sense the upper range of pitches but not the lower tones. Lower tones are sensed by the rate at which the cell fire. We sense pitch because the hair cells fire at different rates in the cornea. And the deafness. So understanding of how hearing works explain hearing problem as well. Conduction deafness occurs when something goes wrong with the system of conducting the sound of the cornea in the ear canal, eardrum, hammer, and so on. For example, my mother-in-law has a medical condition that is causing her steer up to deteriorate slowly. Eventually, she will need surgery to replace the bone in order to hear well. Nerve deafness occurs when the hair cells in the cornea are damaged, usually by loud noise. If you have ever been to a concert, football game, or other event loud enough to leave your ears ringing, chances are you came too close or did cause permanent damage to your hearing. So be careful about like very loud sound. Prolonged exposure to noise that loud can permanently damage the hair cells in your cornea, and these hair cells do not regenerate. So nerve deafness is much more difficult to treat since no method has been found that will encourage the hair cells to regenerate. So this is the explanation of deafness. And the other sensory is touch. When our skin is indented, pierced, or experience a change in temperature, our sense of touch is actively by this energy. We have many different types of nerve ending in every patch of skin. An exact relationship between these different types of nerve ending and the sense of touch is not completely understood. Some nerve endings respond to pressure, while others respond to temperature. We do know that our brain interprets the amount of indentation or temperature change as the intensity of the touch from a light touch to a hard blow. We also sense placement of the touch by the place on our body where the nerve endings fire. Also, their endings are more concentrated in different parts of our body. If we want to feel something, we usually use our fingertip and an area of high nerve concentration rather than back of our elbow, an area of low nerve concentration. If touch or temperature receptors are stimulated sharply, a different kind of nerve ending called pain receptors will also fire. Pain is a useful response because it warns us of potential dangers. Gate control theory helps explain how we experience pain the way we do. Gate control theory explains that some pain messages have a higher priority than others. When a higher priority message is sent, the gate swings open for it and swings shut for a low priority message, which will not fill. Of course, this gate is not a physical gate swimming in the nerves. It's just a convenient way to understand how pain messages are sent. When you scratch on each, the gate swings open for your high-intensity scratching and shut for the low-intensity itching, and you stop the itching for a short period of time. Endorphin or pain-killing chemicals in the body also swing the gate shut. Natural endorphin in the brain, which are chemically similar to opiates like morphine, control pain. The next one is chemical senses, taste. The nerves involved in the chemical senses respond to chemicals rather than to energy, like light and sound waves. Chemicals from the food we eat are observed by taste buds on our tongue. Taste buds are located on papillae, which are the bumps you can see on your tongue. Taste buds are located over the tongue and some part of the inside of the cheek and the root of the mouth. Human sense 5 different types of taste: sweet, salty, sour, bitter, and umami, savory, or meaty taste. Some taste buds respond more intensely to a specific taste and more weakly to others. People differ in their ability to taste food. The more densely packed the taste bud, the more chemicals are observed, and the more intense the food is tasted. You can get an idea of how densely packed taste buds are by looking at the papillae on your tongue. If all the bumps are packed tightly together, you probably taste food intensely. If they are spread apart, you are probably a weak taster. What we think of as the flavor of food is actually a combination of taste and smell. This is our tongue. So here, this is papillae, and then there is a taste bud. And next one is smell. So there is a nose, and go through here. There is two kinds of the olfactory bulbs and olfactory cell and olfactory system. Smell or olfaction. Our sense of smell also depends on chemicals emitted by substances. Molecules of substances Hot chocolate, for example, rise into the air. Some of them are thrown into our nose. The molecules settle in a mucous membrane at the top of each nostril and are absorbed by receptor cells located there. The exact type of these receptor cells are not yet known, as they are for taste buds. Some researchers estimate that as many as like 100 different types of space receptors may exist. These receptor cells are linked to the olfactory bulb, which gathers the message from the olfactory receptor cells and sends this information to the brain. Interestingly, the nerve fibers from the olfactory bulb connect to the brain at the amygdala and then to the hippocampus, which make up the limbic system, responsible for emotional impulses and memory. The impulses from all the other senses go through the thalamus first, before being sent to the appropriate cortices. This direct connection to the limbic system may explain why smell is such a powerful trigger for memories. And next one is body position senses. Our vestibular sense tells us about how our body is oriented in space. Three semicircular canals in the inner ear give the brain feedback about body orientation. The canals are basically tubes partially filled with fluid. When the position of your head changes, the fluid moves in the canals, causing sensors in the canals to move. The movement of these hair cells activate neurons, and their impulses go to the brain. You have probably experienced the nausea and dizziness caused when the fluid in this canal is agitated. During an exciting roller coaster ride, the fluid in the canals might move so much that the brain receives confusing signals about body position. This causes a dizziness and nauseous reaction. And next
So
one,
it's triggered. It's triggered also when you're drunk.
yes, yes,
You feel
right,
dizzy.
yeah. So, hmm. And next one is kinesthetic sense. Kinesthetic sense. While our vestibular sense keep track of the overall orientation of our body, our kinesthetic sense gives us feedback about the position and orientation of specific body parts. Receptors in our muscles and joints send information to our brain about our limbs. This information combined with visual feedback So let's keep track of our body. You could probably reach down with one finger and touch your kneecap with a high degree of accuracy because your kinesthetic sense provides information about where your finger is in relation to your kneecap. See the following table for a summary of senses and their associated receptors. And next one is perception. As stated before, perception is the process of understanding and interpreting sensation. So, psychophysics is the study of interaction between the sensation we receive and our experience of them. Researchers who study psychophysics try to uncover the rules our mind uses to interpret sensations. We will cover some of the basic principles in psychophysics and examine some basic perceptual rules for vision. And threshold. Research shows that while our senses are very acute, they do have their limits. The absolute threshold is the smallest amount of stimulus we can detect. For example, the absolute threshold for vision is the smallest amount of light we can detect, which is estimated to be a single candle flame about 30 miles away on a perfectly dark night. Most of us could detect a single drop of a perfume a room away. Actually, the technical definition of absolute threshold is the minimal amount of stimulus we can detect 50% of the time. Because researchers try to take into account individual variation in sensitivity and interference from other sensory sources. Stimuli below our absolute threshold is said to be subliminal. Some companies claim to produce subliminal message media that can change unwanted behavior. Psychological research does not support their claim. In fact, a truly subliminal message would not, by definition, affect behavior at all because of if a message is truly subliminal, we do not perceive it. Research indicates some messages called subliminal can sometimes affect behavior in subtle ways, such as choosing a word at random from a list after the word was presented subliminally. Evidence does not exist, however, that more complex. So here, there is a box and hearing, touch, taste, smell, and vestibular sense, kinesthetic sense. These are chemical senses, taste and smell, and these are body position senses. And hearing, just like hair cells connected to the organ of corti. And touch, it is temperature, pressure, pain nerve ending. And taste and smell. Taste sweet, sour, salty, bitter, and this one, both. And smell, receptors connected to the olfactory bulbs. And vestibular sense, hair-like receptors in 3 semicircular canals. Kinesthetic sense, receptors in muscles and joints.
Mm.
So subliminal messages such as lose weight or increase your vocabulary are effective. If these tips to change behavior, the change most likely comes from the placebo effect rather than from the effect of subliminal messages. So, if we can see a single candle 30 miles away, would we notice if another candle was lit light next to it? In other words, how much does a stimulus need to change before we notice the difference? The difference threshold defines the change. The difference threshold, sometimes called just noticeable difference, is the smallest amount of change needed in a stimulus before we detect a change. This threshold is computed by Weber's law, named after psychophysicist Ernest Weber. It says that the change needed is proportional to the original intensity of the stimulus. The more intense the stimulus, the more it will need to change before we notice a difference. You might notice a change if someone adds a small amount of cayenne pepper to a dish that is normally not very spicy, but you would need to add much more hot pepper to 5 alarm chili before anyone would notice a difference. Further, Weber discovered that each sense varies according to a constant, but the constant differs between the senses. For example, the constant for hearing is 5%. If you listen to a 100 decibel tone, the volume would have to increase to 105 decibels before you notice that it was any louder. Weber's constant for vision is 8%, so 8 candles would need to be added to 100 candles before it looked any brighter.
Mm.
Next one is perceptual theories. And psychologists use several theories to describe how we perceive the world. First one is signal detection theory. Real-world examples of perception are more complicated than controlled laboratory perception experiments. After all, how many times do we get the opportunity to sit at a single candle flame 30 miles away on a perfectly clear, dark night? Signal detection theory investigates the effects of the distractions and interference we experience while perceiving the world. This area of research tries to predict what we will perceive among competing stimuli. For example, will the surgeon see the tumor on the CAT scan among all the irrelevant shadows and flaws in the picture? Will the quarterback see the one open receiver? In the end zone, despite the oncoming